Let's now have a look at the organization of the nervous system. This is Lecture 5, Section 3. All vertebrate nervous systems are organized into two parts. A central nervous system, which consists of the brain and spinal cord, and the peripheral nervous system, which connects the central nervous system to the rest of the body. All nerves that have their cell body, or if you prefer the soma, located in the brain case or in the spinal cord are part of the central nervous system. In contrast, all nerves which have their cell body or soma outside of the brain case or spinal cord are part of the peripheral nervous system. That's how you determine whether a nerve is part of the central nervous system or the peripheral nervous system. It depends on the location of its cell body or soma. Okay. We can think of the divisions of the nervous system in a very modular fashion, as shown here on this slide. So the CNS, or central nervous system, is positioned to control the physiology and the behavior of an animal. It receives all of the afferent sensory information from the peripheral nervous system. So afferent with an A brings sensory information from the PNS to the CNS. In the CNS, the information is processed and integrated, and then it sends efferent signals, with an E, efferent signals, to cause an intentional and coordinated response via motor pathways in the peripheral nervous system. Motor pathways include the autonomic nervous system and the somatic nervous system, or if you prefer, the involuntary and voluntary responses of the output pathways, or efferent pathways. The sympathetic and parasympathetic branches of the autonomic nervous system work in opposition to each other. For example, in the sympathetic nervous system, the release of adrenaline can cause the heart to accelerate. In contrast, in the parasympathetic nervous system, the release of the neurotransmitter acetylcholine will cause the heart to slow down. So those two branches of the autonomic nervous system counter one another, or work in opposition to each other. Okay, so now you basically know that neurons are connected and they pass electrical signals through these connections via chemical signals, neurotransmitters. The electrical signals within neurons are carried by the charges of ions, right? And when a cell is at rest, it has a resting membrane potential. This is no different than any other cell in the body. All cells have a resting membrane potential. The resting membrane potential is generated by the difference in charge on either side of the membrane in neurons, just like any other cell. The charge on the inside of the membrane is lower than that on the extracellular side, so this causes the membrane potential to be negative. If there was no difference in membrane potential, or I should say if there was no difference in charge across the membrane, then the potential would be zero, as the charges on either side of the membrane would cancel each other out. Okay. The further you get from zero at the level of the membrane in terms of potential, the greater the electrical potential, right? So here we see that the resting membrane potential in a neuron is around minus 70. The greater that value, so the more negative the resting membrane potential is, the more work can be done by this difference in potential across the plasma membrane. And in most animal cells, the resting membrane potential varies quite a bit. It varies between about minus 5 millivolt all the way to about minus 100 millivolt. Okay, so now when a neuron is excited, there's a change in membrane potential. Typically, the membrane potential moves towards zero, and the decrease in charge is used to do something. And that something in a neuron is to initiate an action potential, that is to make the nerve fire. In this figure, you can see that there is a reduction in membrane potential, and we call that a depolarization, as the membrane potential moves from resting membrane potential towards a less negative value, or towards zero. After this depolarization, the membrane potential can be restored back to resting membrane potential. That's called repolarization of the membrane potential. Cells can also be hyperpolarized due to the activation of different membrane transporters. So, for example, depolarization, right, could be caused by the influx of sodium into neurons down their electrochemical gradient, because they're more concentrated. Sodium is more concentrated outside than inside. That could cause a depolarization. A hyperpolarization could be caused by potassium moving down their electrochemical gradient. So potassium is more concentrated inside a cell than outside. If potassium moves out of a neuron, it's going to hyperpolarize the resting membrane potential of that neuron. We're going to spend a considerable amount of time in the next few lectures trying to understand the specific mechanisms that account for voltage differences seen across the cell membrane, specifically, the ion channels that are involved in the depolarization, repolarization, and hyperpolarization states of neurons.